Hello and welcome to tutorial 24. Our topic today is enzymes. Before we get started, just a quick reminder that the notes to accompany this video tutorial are available online through my website. And with that said, let's go ahead and get started. So enzymes are biological catalysts. So I think that the most logical place to start today is with a review of catalysts, which is something that we discussed in more detail way back in tutorial number 14. So what is a catalyst? A catalyst is a substance that speeds up the rate of a chemical reaction without being consumed in the reaction. So let's write that definition up here. A substance that speeds up the rate of a chemical reaction without being consumed. And so you might recall that we traditionally write a catalyst on the arrow in a chemical reaction because it's not a reactant, it's not being consumed, it's not a product, it's not being produced, it's something that is going to be left intact in its original form at the end of the reaction. Okay, so for example, we've seen a lot of reactions written out that require a catalyst, but we'll just use one example. We'll use hydrogenation. Remember that the hydrogenation of an alkene requires a metal catalyst in order to proceed at a reasonable rate. So something like palladium is very common to use in a hydrogenation reaction of an alkene. So I'll just do a general alkene here, adding hydrogen across the double bond. The catalyst, I'll use palladium, will be written on the arrow, and then our product here, when hydrogen adds to each side, is an alkane. Okay, so again, the catalyst is not a reactant, it's not consumed in the reaction, it's not a product, it's not produced in a reaction. So we write it on the arrow to show that it is neither a reactant nor a product, it is a catalyst. All right, so how does a catalyst work? How does a catalyst help reactant be converted into a product at a faster rate? Well, you might remember that every reaction requires a certain amount of energy in order to proceed, and we call that amount of energy the activation energy. And we showed activation energy in tutorial 14 with very general energy diagrams. We were not quantitative about it at all, we were very qualitative, but in general we can show a reaction where reactant is converted to product, and I'll make it an exergonic reaction just because. So this will be our reactant, this is our product, this is the reaction progress, and this is showing the energy. Okay, and that barrier, let's use the pink pen here, this energy that must be reached in order for the reaction to occur is the activation energy, abbreviated as E sub A. So I'm going to write activation energy. And again, we're being general here, just like we were in tutorial 14. We're not specifying how much energy, we're just saying every reaction has some amount of energy required in order to proceed. Some reactions have a larger activation energy barrier, some have a smaller activation energy barrier, but every reaction requires some energy input to get old bonds to start to break apart so that those new bonds can form and the reaction can proceed. So a catalyst works by lowering this activation energy barrier for a reaction. Okay, so we can redraw, and let me go to the blue pen here. The same reaction, but with a catalyst, so the reactants are the same, the products are the same, but the activation energy barrier is going to be lower. So this is a catalyzed reaction. So again, a catalyst works by lowering that activation energy barrier for a reaction so that now even more gentle collisions between reactant particles can possess enough energy to get over the activation energy barrier, which is now lower, and the reaction will then proceed at a faster rate. Okay, so back to enzymes. We said that enzymes are biological catalysts. So enzymes catalyze all the different biochemical reactions that are required to keep an organism alive and well. So let's go ahead and erase here and move on to more specifically talking about enzymes. Enzymes are almost always globular proteins. There are some exceptions to that, but they are few and far between. So let's write up here, enzymes are almost always globular proteins. And the name of an enzyme will usually end in ASE. So it's easy to recognize the enzyme in a reaction. The name will almost always end in ASE. And the prefix to that will typically tell you what substrate, that's just a fancy way of saying what reactant, the enzyme acts upon. Okay, so let's write here, the prefix will tell you what substrate. And again, that's just a fancy word for what reactant the enzyme acts upon. Now, I should just mention really quickly before we go on, there are some old enzyme names that are still in use that don't follow this rule. They don't have the ASE ending, and their prefix doesn't necessarily tell you anything about the substrate that they act upon. But again, those are few and far in between. Most modern enzyme names will have the ASE ending, which makes them nice and easy to recognize, and the prefix that tells you about the substrate that they're acting upon. So for example, let's give a couple examples here. Sucrase is the enzyme that catalyzes the hydrolysis of the glycosidic bond in sucrose during digestion, breaking it down into the monosaccharides glucose and fructose, which can then be absorbed in the bloodstream and be utilized to obtain energy. Lactase, you probably already guessed, that's the enzyme necessary to break down the disaccharide lactose. And some people are lactose intolerant. That means that they are deficient in the use of this enzyme lactase. So they're not able to hydrolyze the glycosidic bond of lactose that links together the galactose and the glucose. So the lactose stays intact, cannot pass into the bloodstream, stays in the digestive system, and causes all sorts of discomfort and bloating and sick feelings, of course, that come along with lactose intolerance. Okay, so hopefully you get the idea. The enzyme names typically end in the ASE, and the prefix gives you some knowledge about what substrate, what reactant the enzyme acts upon. Now, the activity of the enzyme is best described by the induced fit theory. So it's right here. We can fit it in. Let's see. Enzyme activity is best described by the induced fit theory. And so I'm just going to summarize here what the induced fit theory tells us. The induced fit theory tells us that an enzyme has a very small pocket known as an active site, which is where the reaction occurs. So remember, enzymes are almost always these big, huge globular protein molecules, but it's actually a very tiny pocket of that globular protein where the reaction occurs, the active site. So let's write that here. Have an active site which is the location that the reaction occurs. And the substrate, remember that's just again our fancy way of saying the reactant, the substrate will be attracted to this active site, and it will get held in that active site by non-covalent attractive forces, as well as sometimes some temporary covalent attractive forces. Now, of course, we know that an enzyme has to remain unchanged at the end of these biochemical reactions. Otherwise, it wouldn't be a catalyst. Remember, catalysts have to be unchanged by the end of the reaction. So we would expect any of these temporary covalent bonds to be reversed before the reaction is over. Okay, so let's write here. The substrate is attracted to the active site and is held in position by non-covalent and temporary covalent interactions involving the amino acid side groups of the protein. And we saw how those different amino acid side groups can be involved in different attractive forces in our last tutorial, tutorial number 23. All right, so with that substrate locked into that active site, the enzyme then undergoes a structural change. And that structural change causes bonds within the substrate to start to weaken so that new bonds can then be synthesized at a lower activation energy, at a lower energy requirement. So let's write that here. The enzyme undergoes a change in its shape that weakens old bonds of the substrate and positions new bonds for formation. And once that substrate is converted into product, the enzyme releases it and the enzyme resumes its original shape and configuration. So it's right here. Enzyme releases the product and resumes its original shape. Okay. So just really quickly, I'll go to a different color over here. Everything we've said here, I'm going to just summarize it with sort of a generic enzyme catalyzed reaction. Okay, so hopefully this will help with all these words, putting them into a better perspective. Okay, so we've got an enzyme. I'll just write E for enzyme. Remember I said I was going to be very general here. And we've got a substrate. The enzyme has an active site, and that substrate is attracted to that active site, and it is held in position in that active site by non-covalent as well as temporary covalent interactions. I'm going to indicate that with this dashed line. Okay, so this dashed line is indicating my interactions that hold the enzyme and substrate together. So let me label that in green just to make it more clear. So interactions hold the substrate in the active site. Of the enzyme. So that's what that dashed line is supposed to be representing there. And again, those interactions can be non-covalent interactions with the amino acid side groups, and they can also be temporary covalent interactions with those side groups. Okay. The enzyme undergoes its structural change, and that substrate is locked in that active site. So it gets stretched, it gets manipulated. Old bonds get weakened so that new bonds can form more readily. Once those new bonds are formed and the substrate has been converted into the product, the enzyme releases that new product and resumes its original conformation. Now it's ready to catalyze another reaction all over again. Okay, let's go ahead and erase here. And we're going to go on to slide number 3 and talk about cofactors and coenzymes, which are commonly needed to make an enzyme function properly. Enzymes often require a cofactor in order to function properly. And a cofactor can be a metal ion, like a copper ion or a zinc ion, or an organic molecule. When a cofactor is an organic molecule, you'll usually hear it referred to more specifically as a coenzyme. And coenzymes often have a vitamin component to their structure. And so you can see how a vitamin deficiency would lead to a deficiency in these particular coenzymes, which are required for an enzyme to function properly. Now, very important note I want to make about cofactors and coenzymes is that they can be changed in an enzyme catalyzed reaction. Remember that an enzyme cannot be changed overall. It can be changed temporarily during a reaction, but it has to be regenerated by the end. Otherwise, it doesn't fit the definition of a catalyst, something that is not used up in the reaction. But a cofactor or coenzyme can be changed. So let's write that up here. Very important. Can be changed in an enzyme catalyzed reaction. So you can see how these cofactors can really lead to more diversity for the enzyme. For example, if the enzyme is responsible for catalyzing an oxidation reaction, the substrate's going to get oxidized. We know that in order for something to be oxidized, something has to get reduced. Those occur together. So it will be the cofactor or the coenzyme that gets reduced. It can't be the enzyme, not permanently anyway. So the coenzymes and cofactors are able to be changed. So in these oxidation and reduction reactions, we're going to see those being either oxidized or reduced. Two coenzymes that we are going to see a lot of when we get to metabolism include nicotinamide adenine dinucleotide and flavin adenine dinucleotide. So I'm going to go ahead and write those up on the board right now. So two coenzymes we will see frequently include nicotinamide adenine dinucleotide, NAD plus for short, and flavin adenine dinucleotide, or FAD for short. And these coenzymes will be involved in redox reactions. So we will see them oscillating between their oxidized and the reduced forms. So I'm going to go ahead and write the reduced forms up here as well. And I'm going to do a double-headed arrow, so I'm indicating that they will be seeing them going both ways. So the reduced form is NADH, an H+. It gains electrons and hydrogen atoms. And the reduced form of FAD is FADH2. Okay, so let me label that. That's very important, or it's going to be very important once we get to metabolism. Over here, I've written the oxidized forms of the two coenzymes, and over here, I have written the reduced forms. For these two coenzymes. So what I'd like to do now is go ahead and zoom in on slide 3, where I have drawn out the entire huge, complicated looking structure for each of these coenzymes in both the oxidized and the reduced form, so you can actually see where that site of reduction occurs. Okay, so zoomed in on slide 3, we can see NAD, the nicotinamide adenine dinucleotide, in its oxidized form, drawn out fully. And I'll just point out real quick that a component of NAD is vitamin B3, better known as niacin, right up here. And that's actually the site of reduction. Notice that your hydrogen and your 2 electrons form right here, form a bond right here. And so we get then the reduced form of that coenzyme, nicotinamide adenine dinucleotide, in the reduced form. Same thing, real quick over here with FAD. We've got the oxidized and the reduced form drawn out fully. And I want to point out real quick that vitamin B2, better known as riboflavin, is a component of this coenzyme. So this part I'm kind of circling here with my finger is vitamin B2. And it too is the site of the reduction, where the 2 hydrogen and the 2 electrons add to give you the reduced form of a coenzyme, the FADH2. Okay, so again, we'll be seeing those coenzymes a lot, so I like to go ahead and show their structure, show the vitamin components to those structures, and show the site of the reduction. So we're going to finish up with slide 4 of the notes, classes of enzymes. Enzymes can be divided into 6 main categories based on the type of reaction that they catalyze. And I've written those 6 categories up here already in no particular order other than this is the order that I wrote them in the notes. So let's go ahead and get started with the first category, oxidoreductase enzymes. Oxidoreductase enzymes will catalyze redox reactions upon substrates. So let's write that up here. Catalyze redox reactions of substrates. And you might recall that when something gets oxidized, something else has to get reduced, and vice versa. So not surprisingly, these enzymes, these oxidoreductase enzymes, will require a coenzyme, which can either be oxidized or reduced. All right, so let's write that up here. Will require coenzyme. And we just took a look at 2 of the more common coenzymes that will be used in these redox reactions. Let's write them up here real quick. Nicotinamide adenine dinucleotide, and I'll write its reduced form as well. And flavin adenine dinucleotide, and I'll write its reduced form as well. And then another one that you'll probably see a lot in your biochemistry studies is very similar to the NAD. It's going to be NADP, the nicotinamide adenine dinucleotide phosphate. So I'll go ahead and write that under here as well. And it's the same structure as NAD that we just looked at on slide 3, except that the sugar component of the molecule has a phosphate unit attached to it. So a very small difference in the structures there. And I'll write its reduced form as well. Okay, so all of those. I'll close my bracket down here. Put a comma there. Okay. All right, so let's go ahead and just show a quick example of an oxidoreductase enzyme catalyzing a reaction. So a good example would be the oxidation, the first step of oxidation of alcohol in metabolism of ethanol. Okay, so let's use ethanol as our alcohol, CH3, CH2OH. And in the first step of its metabolism, it gets oxidized into the aldehyde ethanal. So CH3 O, and an H. And the enzyme responsible for this step is called alcohol dehydrogenase. So let me write the enzyme name, alcohol dehydrogenase. Notice the ASE ending, and the term alcohol tells you it's going to act upon an alcohol. It doesn't have to be ethanol, but that's traditionally the alcohol that we ingest, right? So we'll do ethanol here. And then we're going to write our coenzyme as a coupled reaction here. We're going to write that this requires the coenzyme NAD being reduced to NADH. H+. Okay, so our coenzyme, the reaction of our coenzyme, the reduction is shown here, the oxidation of our substrate here, and then our enzyme name on the arrow. Okay. All right, so there are different subcategories within each of these 6 categories. So for oxidoreductase, you will see names like, and I'll write them over here to the side, I'll write them in green so they stand out a little bit. You'll see names like oxidase at the end of the enzyme name. That means that this is an enzyme that is oxidizing, specifically oxidizing a substrate. You will see the term reductase. That's referring to an enzyme that specifically is reducing a substrate. And we will also see a lot of dehydrogenase enzymes. And that's an enzyme that is oxidizing a substrate specifically by the removal of hydrogen, like what we saw here. Okay. Going on to transferase enzymes. Go back to the blue pen here. A transferase enzyme catalyzes the transfer of a group from one molecule to another. So let's write there. Write that up here. Catalyze the transfer of a group from one molecule to another. And common subcategories of transferase enzymes include transaminase enzymes, which specifically are transferring an amino group from one molecule to another, and kinase. Kinase enzymes transfer a phosphate group from one molecule to another. All right, category number 3. Let's go back to the blue pen. Hydrolase enzymes catalyze hydrolysis reactions. So we're going to be seeing a lot of these when we get to digestion. So let's write up here, catalyze hydrolysis reactions. And we already know that hydrolysis is referring to the breaking apart of a bond, the splitting apart of a bond with water, with H2O. So we're going to see a lot of lipase enzymes hydrolyzing our lipids in metabolism, or in digestion, I should say. We're going to see protease enzymes hydrolyzing our peptide bonds in proteins during digestion. We're going to see very specific enzymes hydrolyzing our glycosidic bonds during the digestion of different carbohydrates. So things like sucrase, which will hydrolyze the bond in sucrose. lactase, which will hydrolyze the glycosidic bond in lactose, amylase, which will hydrolyze the glycosidic bonds in starch. So let's write over here in a green pen some common ones that we're going to see. Again, this will mostly be during digestion, will be the lipase enzymes, the protease enzymes, and then different specific enzymes for the glycosidic bonds like sucrase, lactase, amylase. Those are all going to be hydrolyzing different types of bonds. Ester bonds, peptide bonds, and glycosidic bonds. All right, so on to category number 4. The lyase enzymes will catalyze the removal of a molecule to form a double bond, or the reverse, the addition of a molecule to a double bond. So let's write here, catalyze the addition of a molecule to a double bond or the reverse. Meaning they catalyze the removal of a molecule to form a double bond. I'll write that in parentheses. The removal of a molecule to form a double bond. Okay, isomerase enzymes, much like the name implies, they catalyze isomerization reactions. Remember that isomers are molecules that have the same molecular formula but a different connectivity of the atoms. So basically an isomerase enzyme just takes the atoms in a molecule and rearranges them somehow. Okay, so I'm going to write up here, catalyze the isomerization of a substrate. So again, just taking the atoms in that molecule and rearranging them and connecting them in a different way, making an isomer. All right. And last but not least, the ligase enzymes will catalyze the coming together of two molecules to make a bond. Okay, so let's write here, catalyze formation of a bond between two substrates. All right. Okay, so we will go ahead and conclude our tutorial 24 right here. This was just an introduction to the topic of enzymes, and hopefully you will consider tuning into problem set 24 so that we can go over some problems involving enzymes. Thank you very much.